Hello and welcome everyone to today's Evo Eco seminar. Before I introduce today's speaker, next week we'll have Anil Agrawal, and we'll soon be announcing the whole schedule for April at least, but you can already see it here on the slide as well. So we have Tracy Chapman, Anjali Goswami, and Pat Monaghan later in April. And with that, today's speaker is Professor Troy Day. Troy started his career at UBC, after which he moved to Queen's University to get his PhD with Peter Taylor. He then did a postdoc with Sally Otto back at UBC. And Troy is currently a professor at Queen's with a cross appointment in the departments of mathematics and statistics, as well as the Department of Biology. As a mathematical biologist, Troy has explored a variety of questions in evolutionary biology, including kin selection, altruism, and aging. You may also know Troy from his book with Sally Otto that teaches mathematical modeling to biologists. And much of Troy's work in the last 20 years or so has focused on the evolution of pathogens and parasites and so-called evolutionary epidemiology, which is what he'll be talking about today. And with that, Troy, the stream is yours.
Thank you. And thanks for the opportunity to give a presentation in this series. And I want to thank the organizers for putting this series together to begin with and keeping it going over the past year. It's been a fantastic way to stay connected with people in this kind of strange pandemic year. And for me, at least, it's given me an opportunity to see a number of great talks that I probably actually wouldn't have had an opportunity to see otherwise. So it's been fantastic. At least there's something good that's come out of it, hopefully. What I'm going to talk about today is the evolutionary epidemiology of emerging infectious diseases. And I do this with a bit of hesitation. I am going to talk about some recent work on COVID-19, but I realize that probably a lot of you are sick and tired of hearing about infectious diseases. What I want to do is present the recent stuff we've been doing kind of in the context of some older work that we've done over the past several years. And I'm going to kind of intermingle it with results of other people as well to kind of try and paint a broad backdrop of how our current disease of focus, how it's emerged and how it's evolving, how that all fits into the broader study of the evolution and emergence of infectious diseases, at least in terms of the kinds of questions that I've been interested in. So my talk is going to have three main parts, and it's going to follow basically the kind of life cycle of an emerging infectious disease. I'll talk a bit about the processes, epidemiological and evolutionary processes involved in the emergence of new infectious diseases. And then I'll move on to some ideas related to how we might expect evolution to operate on pathogen populations once a disease has taken hold in a new host population. So as it's spreading through in an epidemic way, or maybe even a pandemic way, how is selection acting? And then as we move towards a more endemic situation, how does the nature of selection change and the expectations of what we might see evolutionarily change over that kind of changing epidemiological demographic? And the very last thing I'll do is talk a bit about some, what kind of ideas might we take from evolutionary biology to help us control infectious diseases like COVID-19. And this is going to be a couple of slides, really something very brief. So let's first talk about the emergence of infectious diseases. And maybe one of the nicest ways to kind of conceptualize this is through this, what's called a pathogen pyramid. And the idea is there's kind of 4 levels or stages that the emergence of a novel infectious disease goes through. And in the first level or stage, this level 1 is basically an exposure stage. So if we're thinking about something like humans acquiring a new infectious disease, first we have to be exposed to some novel pathogen source. And typically we're thinking of cross-species transmission from an animal reservoir. There are other possibilities, but that's the main thing people tend to think about. Most of the time, those exposure events probably don't result in much happening, but there's some subset of those exposure events that can result in infection. And so that's stage or level 2, and individuals might become infected, but the idea in level 2 is although these infections might even be quite severe, there's not necessarily any onward transmission within the novel host population. And so to move to level 3, there has to be some level of host-to-host transmission in the new host population. And the idea in level 3 is that there is transmission, but it's still limited to some extent. And the infectious disease is ultimately destined to still die out. So we might have small kinds of chains of transmission, but still no epidemic spread. And it's only once we reach level 4 that we get epidemic spread and potentially pandemic spread. And kind of in the jargon that everybody knows about now, the distinction really between level 2 or level 3, sorry, and level 4 is level 3, we have an infectious disease that has an R0 value that's less than 1, so it can spread, but it's not going to actually take hold and have a self-sustaining transmission cycle. Whereas those instances in level 4, they have an R naught that's bigger than 1. And so they can potentially at least spread and be maintained in the new host population. Now, there's a lot of interest, there has been a lot of interest, and much of it actually stems from the SARS outbreak in 2003 and trying to identify risk factors for the emergence of infectious diseases. There's a huge amount of work that's been done in that area, and I'm just going to kind of skim over the surface of it, some of the empirical stuff and some of the theoretical stuff. This is a study by Mark Woolhouse and colleagues, and they've done a lot of the nice foundational work in this area. And what they were interested in doing is, so they basically collated a number of infectious diseases of humans, and they categorized them as to whether or not they were associated with emerging infectious diseases or not. So a suite of different pathogens, some of them seem to be associated with as emerging infectious diseases, and others have been there for a while. And then they went through and also identified where in those instances of emerging infectious diseases did the infectious agent come from in terms of an animal reservoir. And this is the histogram of what they have in that particular data set, at least. And there are a couple of things I want you to take away from this histogram. First, although bats typically get a bad name in terms of being a reservoir for infectious diseases, they're certainly important in this analysis, but they're by no means the only animal reservoir source of novel infectious diseases, at least in this analysis. The other thing to notice is that most of these species are mammals, right? And that's maybe not surprising when we're talking about infectious diseases of humans, because we might expect that we're going to acquire most of our novel infectious agents from other species that are at least to some extent closely related to us. And that kind of question has actually been analyzed in quite a bit of detail as well. And I'll just show you briefly some results from a study by Davies and Amy Peterson that looks at this kind of question of the evolutionary relationship among host species. So what they did is they asked the question, do host species that tend to be closely related also tend to share pathogen species? And so they took a data set of over 100 wild primates in this case, and I think over 400 different infectious agents of those primates. And then they looked at all pairwise comparisons of the primates and asked, is there a relationship between the similarity, or how is the similarity in the pathogen assemblage between these two primate species related to their evolutionary divergence? So on this graph, the vertical axis here is the degree of similarity in the pathogen assemblage between a pair of primate species. On the horizontal axis is the divergence time between that pair of primate species. And the data are binned on the horizontal axis into bins of 10 million years. And the take-home message here is there's this quite strong relationship between the two. So host species that tend to be very closely related evolutionarily tend to share a lot of similar pathogens compared to those that are more distantly related. Again, sort of suggesting this idea that maybe the most dangerous animal reservoirs are those that are most closely related to us. You can see the data is separated by sympatry and allopatry as well. Does the pair of primates in question co-occur or not? And you can see there's a signal of that in the data as well. Another thing you can do, and this is a couple of really kind of foundational papers as well by Cleveland et al. And Taylor et al., is you can start to ask questions about how the nature of the parasite itself affects evolutionary emergence. And so in this first data set, what they did again is take a sort of collated a number of pathogens of humans, and again categorize them as to whether or not they were associated with emerging infectious diseases. And they also classified them by their mode of transmission, vector-borne, direct, or indirect, and then computed a relative risk. And so the idea here is if the relative risk is bigger than 1, then pathogens with that transmission mode tend to be associated with emerging infectious diseases more than you'd expect based on their relative abundance. And so you can see in this analysis, at least, vector-borne pathogens tend to be associated with emerging infectious diseases more than you might expect based on their abundance. And the opposite is true of indirectly transmitted pathogens. So that's one kind of analysis you can do. And again, these papers have done a number of analyses. I'm just kind of skimming over the surface of some of the things that are most important for what I want to talk about. You can also classify in that same data set the pathogens by their taxonomic group. So whether they're viruses, protozoa, and so on. And again, compute this relative risk. And what you see, and it's a very robust pattern in a number of other analyses as well, is that viruses in particular tend to be disproportionately associated with emerging infectious diseases compared to what you'd expect based on how abundant they are. And the opposite is true for fungi and helminths. They tend to be disproportionately not associated with emerging infectious diseases based on how common they are. Now you can dig a little deeper into this data set. In particular, if you look at viruses, you can subdivide them into whether or not they're RNA viruses or DNA viruses. And if you do that, there's a pretty decent signal showing that it's RNA viruses in particular that seem to be disproportionately associated with emerging infectious diseases. And you can think of a number of reasons why that might be true. Maybe it's just something as simple as them having a higher mutation rate, and so maybe they're more adaptable to new hosts. And this great book by Eddie Holmes here goes through that and many other things about the evolutionary genetics of RNA viruses if you're interested in that topic. So that's just a skim of some of the empirical work. As I said, there's a lot more if you're interested in that sort of thing. I'll say a bit about theoretical work that's been done along these lines as well. And maybe the simplest way you could model this theoretically is imagine dropping a new pathogen into a new host population. Then you model the contacts among those host individuals. And every time there's a contact, there's some probability that that pathogen gets transmitted to a new individual, that probability being T here. And then if you run a model like that, what you find is this sort of branching process of transmission. So in this plot here, the idea is this is the first person or individual, whatever the host species might be, that's infected. By chance, it's generated 2 new infections. This one has only generated 1 more, and so the chain goes on. This one has generated 2, and so on. But there's a lot of stochasticity in this initial stage of the spread. So this chain of transmission typically very often goes extinct. But every so often it takes hold and it results in an epidemic and potentially a pandemic spread as well. And you can take models like this and compute what the probability of that establishment is. And there are many models that do this sort of thing, going back at least to this paper by May and Lloyd. What I'm going to present here are some results by a former master's student in my group, Helen Alexander, who's now a prof at the University of Edinburgh. And what she did was build a model that sort of had this kind of flavor to it, but she explored a number of different things in terms of contact structure and that kind of thing. And I'll just show you some simple results from some of the analyses she did. So this, you can compute this quantity R0, which tells you something about the probability of emergence of the disease. And what you find is there are 3 critical parameters that tend to appear. One of them is the transmissibility of the pathogen from one host to another. And of course, that as that goes up, the probability of establishment goes up. And so when we're thinking about evolutionary relationships among hosts, if you acquire a pathogen from a closely related reservoir species, maybe this T is going to be larger than it would be if you acquired a pathogen from a more distantly related species, because it's already to some extent adapted to the new host species. Another important parameter is the average number of contacts. Not surprisingly, as that goes up, the probability of establishment goes up as well, because there's more opportunities for transmission. And maybe slightly less obviously, the coefficient of variation in the number of contacts individuals have has a positive effect on establishment. In particular, the more heterogeneity there is in the population, the greater the degree of variability among individuals in the number of contacts they have, the greater the probability of establishment. And the reason is basically that when you drop a pathogen into this population, if there are some individuals who are highly connected and some that have very low levels of connection, very quickly the pathogen will be associated with those nodes that are highly connected by virtue of the fact that they're highly connected. And so they will then tend to transmit the pathogen onward by their high numbers of connections in a way that's greater than the average in the population. And that enhances the probability of establishment. So you can see in this plot, this is T along the horizontal axis, the transmissibility of the pathogen variant, and this is probability of establishment on the vertical axis. Populations of hosts that have a high level of heterogeneity in the number of contacts can support the emergence of pathogens that have lower levels of transmissibility than populations that are homogeneous, the Poisson case in this instance. So that's one model for how we might get the emergence of an infectious disease. The pathogen is already adapted enough to the new host population that it can cause an epidemic. Another possibility is that there has to be some level of evolutionary adaptation in the new host species before it can cause an epidemic. And really, I think the first analysis of this in a comprehensive way was this nice paper by Antia et al. In 2003. And the idea is, again, you get a animal reservoir transmits a pathogen to the new host population, and there may be some level of spread. So we're in that kind of level 3 in the pathogen pyramid, but R0 is less than 1, pardon me, less than 1. But you get mutations appearing during this chain of transmission. And if a mutation appears that causes it to be more transmissible, like in the red case here, that can then spark a self-sustaining outbreak that goes on to be an epidemic or a pandemic. So there's this sort of evolutionary emergence process. And Helen analyzed this as well in the context of that kind of model where we have contact networks, and she looked at different kinds of mutational networks among genotypes of pathogens. And again, I'll just briefly mention one of the results that she derived. So again, you can see there's this nature of the contact distribution matters in the probability of emergence. But now it also matters what the different transmissibilities of the different genotypes of pathogens are. And this U here is a matrix that accounts for the different ways the genotypes mutate amongst one another, and how selection within an individual host can drive the frequency of these different genotypes up or down. And one of the things Helen was interested in looking at was this question about mutation rate. So if it is the case that RNA viruses are more often associated with emergence than we would expect based on their abundance, because of an enhanced mutation rate, we can explore that theoretically. And of course, you need to make some assumptions to do that, and we can talk about those later if people are interested. But here's a particular example of the kinds of results she found. So mutation rate on the horizontal axis, probability of emergence on the vertical axis, and these different lines correspond to different levels to which the initial variant is adapted to the host population. So how good is this initial variant that comes in here? How good is it at transmitting before any evolutionary adaptation happens? And you can see for pathogens that are initially poorly adapted in their host population, increasing the mutation rate does a lot to increase the probability of emergence of the infectious disease. But that effect decreases as the initial level of adaptation increases. And then there's a threshold above which, if the pathogen can actually sustain itself in the new host population without any adaptation, then we get a reversal in the relationship. Increasing the mutation rate can actually be detrimental to the probability of emergence. So it really does depend, at least on the assumptions here, on the level to which the pathogen is initially adapted to the new host population in terms of the effects of mutation rate on the probability of emergence. So we can talk a bit about SARS-CoV-2 in this context. So we can ask, where did it come from? When did it arise? And I'll say just a little bit about some analyses that have been done along those lines. This is some really nice analysis that was done very quickly during the early stages of the pandemic. We now, as far as we can tell, think that it arose as a result of a cross-species transmission, probably from bats, but maybe also involving some other intermediate species like pangolins. There's also been some nice analysis, and this was published just a couple of weeks ago in Science, on the time when we might have expected this thing to emerge. And so what was the date of the index case, the very first person to have been infected, basically? And one way you can try and analyze that is by taking a sample of pathogen genotypes at a particular time. So let's say these guys right here, and then using coalescent techniques, tracing backwards in time to try and find the date at which the most recent common ancestor occurred. And that's sort of this big dot right here. Now, of course, that date when that most recent common ancestor occurred need not be the date when the index patient was first infected, the very first infection, because remember, I showed you that during the initial stages of an outbreak, you get this highly stochastic process where there are lots of branches of transmission that ultimately go extinct. And there maybe is only a single trunk that ultimately gives rise to the eventual pandemic. And so there can be this window of time between when the coalescent event happens in the analysis and when the index case was actually, when it actually occurred. So in this paper, what they did is built branching process models like the ones I just described and simulated forward, took that simulated data, and then they applied coalescent techniques and tried to infer the time when the most recent common ancestor was. So they'd infer the date of this point here. And because it was simulated data, they also knew the date of the very first infected individual. And they could then get an estimate of this window of time between when the index case happened and when this coalescent event happened. And so sort of tying this into data on SARS-CoV-2, they were able to come up with a point estimate of the index patient's infection date around the beginning of November. Pretty big window of error around that, but it was a pretty robust analysis in terms of the different kinds of things they looked at. They also noted that the vast majority of these simulations that they ran resulted in extinction. And so, in other words, the pandemic event that we're living through right now was by no means guaranteed. If you replayed the thing over and over again, the claim is that the vast majority of those would never have taken hold. And the ones that did tended to be those, like in the general theory, where there was a high average number of contacts among individuals, and there was a high degree of heterogeneity in the number of contacts in the population. Okay, so that's just a very quick, brief tour through some ideas and processes involved in the emergence of infectious diseases. I'm going to move on now to talk about how we might expect evolution and adaptation to operate as we move through a pandemic phase or an epidemic phase into an endemic phase. And I'm going to do this first by talking about a very simple model for a respiratory disease. What I'm going to talk about generalizes to more complicated models, as we'll see. In this simple model, I'm just going to imagine there's a pool of susceptible individuals, and these individuals can be infected by one of any number, let's say n different pathogen strains. And the pathogen strains will determine the transmissibility. So pathogen strain I has transmissibility beta I. And the genotype of the pathogen also determines the virulence, the extent to which infection kills the host. That's this V sub I here. And then individuals can recover from infection as well. And so you can build a simple ODE model where we're tracking the number of, or the density of susceptible hosts. There's an inflow, there's an outflow through natural mortality, and there's an outflow through infection that depends on the average transmissibility across all genotypes of the pathogen in the population. That's this beta bar. And then the total number of infections changes through time as a result. Well, there's an inflow through infection at this average rate beta bar. And there are three ways to lose an infection: natural mortality, you can clear the infection, or you can die from the infection. And again, at a rate that on average depends on the average virulence of the genotypes in the population. Okay, now what we're typically interested in a model like this, or what I have been interested in, and a number of the people I've been working with, are the evolutionary dynamics of beta bar, the average transmissibility, and the average mortality that the pathogen induces. And this is work that started quite a number of years ago, first with Steve Prue, who was a postdoc in my lab and is now at UCSB, and then over a number of years, an ongoing collaboration with Sylvain Gandon. And I'll just describe the dynamics in this particular simple model. So this equation in the box here, looking at this top one, this tells us how we expect the average transmission rate to evolve over time. There's two components to it. The first component has the genetic variance in transmission rate multiplied by the strength of selection on transmission. And S here doesn't stand for strength of selection. S is the number of susceptible hosts. So the important point here is that the strength of selection on transmission is proportional to the density of susceptible hosts in the population. Enhanced transmissibility is more beneficial to a pathogen if there are lots of individuals that it can actually have access to. And then the second term is any indirect effect due to selection on virulence. So virulence is always selected against. It's always bad to kill your host because it shortens the period of time over which you can transmit. And so if there's any genetic covariance between transmission and virulence, that will impose an effect on the evolution of transmission as a result of this indirect effect of selection. And there's an analogous equation for V bar. Maybe it's more familiar to many of you if you write it in matrix form, which is given here. So it's like the multivariate Breeder's equation or Landy's equation. So we have a vector of the evolutionary change in the average value of the traits is equal to the genetic variance covariance matrix of the pathogen population times the vector, the selection gradient vector. And we can conceptualize it geometrically by plotting different pathogen genotypes. Those are these different dots here in the plane where we have transmission rate on the vertical axis and virulence on the horizontal. And so that blue dot is meant to be the average value of each of these traits. And selection is always pushing the average virulence downwards, and it's always pushing, it's always pushing the average transmission rate upwards. And so the direction of evolution will depend on the vector of the selection gradient as well as the nature of genetic variation that's present. Now, in the case of COVID-19, maybe we're more interested not in a whole suite of variants, but just two variants, like the UK variant, for example, and a wild type. And so I'll just say a bit about how things play out in that more simplified case. And this is actually a set of ideas that dates all the way back to this nice paper by Lenski and May. And so let's imagine we have a wild type in blue here, and we've got a mutation that has an enhanced transmission rate, but also an enhanced virulence. And during the early stages of an outbreak, during the pandemic phase of an outbreak, there's going to be very strong selection for increased transmission because there's a lot of susceptible individuals around, this S right here. And so this part of the selection gradient is very strong. And so we might expect the frequency of this more transmissible variant to increase, despite the fact that it having higher virulence is detrimental to its fitness. The gain it has through this enhanced transmission more than outweighs the cost it pays from enhanced virulence because of this huge abundance of susceptible hosts. And then as we move into the endemic phase of the outbreak, the density of susceptible hosts will go down, and so the strength of selection on transmission will go down. And we might expect an evolutionary reversal where now the wild type increases in frequency because it's gaining from the reduced virulence, and there's not that much it can gain from having an enhanced transmission since there are very few susceptible hosts around. So that's kind of the general idea. Now, this idea was actually tested by Sylvan's group, and this is a paper by Berngruber et al. In PLOS pathogens, and it was tested in a system of lambda-phage and E. coli. So I'll just quickly tell you a bit about the life cycle in this experiment. So E. coli is the host in this experiment, and lambda-phage is the pathogen. And the life history of lambda-phage is one where when it infects E. coli, it can either integrate into the host genome, and it can replicate vertically as the host replicates, or it can lyse the host, burst the host, killing the host and producing a bunch of propagules that can then transmit horizontally. And in this experiment, Berngruber had two different genotypes of the pathogen. One was a sort of low virulence pathogen, so it tended to go into this integration pathway, and very seldom did it kill the host. And as a result, very seldom, or it wasn't very good at transmitting horizontally. And the other mutant genotype was the opposite. It much more often burst the host, so it had a high mortality rate for the host, but it also had a very high horizontal transmission potential by doing that. And so they conducted an experiment basically competing these two against one another under different epidemiological scenarios. And I'll first present a model that they built to try and make predictions kind of along the lines that I just showed, but in a very quantitative way for this specific system. So the blue line here corresponds to an endemic experiment. So they started the experiment where the prevalence of infection was very high, and so it kind of just remained high. The red line is a case where they started with a low prevalence. So there was this initial epidemic phase to the outbreak, and then it reached this endemic state. At least in the model, that's what happened. And then in that model, they also asked what happens to the relative frequency of the virulent type. That's what's shown at the bottom here, starting at 50-50. So in the case where they looked at the endemic experiment only, the virulent type just monotonically decreased in relative abundance over time. In the case with this epidemic phase that happens, what happened in the model is you get this transient evolutionary increase in the type that's more transmissible but more virulent, for exactly the reasons I described a minute ago. And then once you reach the endemic phase, it starts to decrease in frequency like that. So they basically ran these exact two experiments in a chemostat with these two genotypes of pathogen. And the results they got are very close to the model predictions that they made. So I won't say too much more about that other than just to note that if you look at the red case, so this epidemic case where there was initially this epidemic spread through the population, you get this transient increase in the more transmissible, more virulent type until endemicity is kind of reached. And then you get this decrease again, although it didn't decrease all the way in a sort of continuing monotonically downward trend like was seen in the model for some reason. So that's a nice experimental verification of those general ideas. Of course, the models that I've talked about so far are pretty simple in the sense that I've assumed that the transmission rate is constant over an infection and that mortality, the virulence, is constant over an infection. And we know that that's not true for almost all infections. So in COVID-19, for example, there's a period of time at the start of the infection where there are no symptoms and no transmission. Then there's a period of time where there are very few symptoms, but there's significant transmission. And then after that is when mortality sets in. And so there's this important aspect of the timing of what you might call disease life history events over the course of an infection. And we can build models to try and explore how we'd expect that timing structure to evolve in the context of epidemic and endemic scenarios as well. And I'll just say very briefly a bit about this because it's quite involved to get into. So I'm going to focus only on the case of transmission rate evolution in these theoretical results. And there's some empirical results I'll show you. But you can incorporate virulence evolution into it in a very similar sort of way. And so this is work that was actually led by Nicole Medeo when she was in my group. She's now a prof at the University of Toronto. And I'll just explain what's going on here with reference to this very simple model that I started with for the evolutionary dynamics of the average transmission rate, but now in the absence of anything to do with virulence. So that average transmission rate evolves in a rate that's proportional to the density of susceptible hosts, as we've seen, and proportional to the genetic variance in the transmission rate. Now we can allow for transmission rate to evolve differently at different ages of infection. And so we get a model that looks like this. So A here is now age of infection, 0 at the beginning of infection and so on. And so this tells us how the average transmission rate at infection age A is predicted to evolve. Again, it's got these two parts. It's proportional to the density of susceptible hosts, but now this genetic variance bit is replaced with this more complicated looking thing, which involves the genetic covariance between transmission at one age of infection and transmission at other ages of infection. Okay, and the structure of this model is basically an extension of this nice set of ideas that Kirkpatrick and colleagues have developed over the years for the evolution of function value traits in quantitative genetics. So this G here is the genetic variance covariance function, measuring the covariance between transmission at different ages during an infection. And Q is the frequency distribution of infections of different ages. Okay, so you can take that kind of model, and what Nicole did was take that model and then take data from, in this case, a model system of malaria in mice. So this was a collaboration with both Andrew Reed's group and Sarah Reese's group, taking data from the blood stage infection part of a malaria infection in mice, and looking at different genotypes of malarial pathogens, P. Gibbodi in this case, as a function of infection age. And so here's the kind of data that Nicole used. So these are 6 different genotypes of pathogen, plotted in their density against infection age. And you can see, you know, there's variability in their so assuming that transmission rate is proportional to this log parasitemia, there's variability in transmission rate throughout the course of infection. And so what Nicole did is take this data and try to estimate this G function. And I'll just show you what the G function looks like graphically. So this is basically looking at a G matrix, but now it's a G function, I guess, if it is the best way to describe it. So this is age of infection on both axes here. If I trace out the diagonal, that tells me the amount of genetic variation that's present at different infection ages. So blue is positive, yellow is going to be negative here. So if I look at early infection ages, 6, for example, there's some degree of genetic variation in transmission rate there. That's reflective of this variability here at early infection ages. If I trace along the diagonal to late infection ages, it looks like there's lots of genetic variation in transmission rate, and that's reflective of this greater degree of variability among the genotypes for transmission rate late in the infection. And interestingly, if you then look at the off diagonal, genotypes that are highly transmissible early in an infection have low levels of transmission late in the infection, and vice versa. So even though transmission is always selected to be higher at all infection ages and in a way that's proportional to the density of susceptible hosts, in this system at least, and at least for these particular mice that it was tested in, there's a trade-off between being able to transmit well early versus being able to transmit late. That's mediated through these different genotypes. And so what Nicole did is take this data, and you can kind of come up with confidence bands on this estimate of the G function, and then run it through this model to make predictions about what you'd expect to happen evolutionarily under epidemic and endemic scenarios. And this is one snapshot of some of her results. So if you look at the epidemic scenario, the age of infection distribution is pushed to young ages. Most infections are very young, and there's lots of susceptible hosts around. And so that places a very strong premium on being able to transmit well early in an infection. But that comes at the cost of being able to transmit late in an infection. So you get the evolution of lots of transmission early and less transmission late. As it moves into the endemic phase, the pattern reverses, and you get the evolution of much higher transmission late in an infection at the expense of reduced transmission earlier on, again, because of that negative genetic covariance. As I said, these kinds of ideas, we can incorporate virulence, right? So there's another trait that would be laid out along age of infection, and we'd have to deal with not only the genetic covariance between virulence at different infection ages, but also the cross covariance between those two. And I'm not going to talk about that. I'll just quickly say how it's one way to think about how we can talk about virulence evolution in the context. And it's related to ideas about the evolution of senescence. So in the evolution of senescence, the main idea is that as you look at traits that are expressed later and later in an organism's life, the strength of selection on them is weaker. And the reason is basically because the organism has undergone most of its reproduction by the time those late ages have happened. And the same kind of thing is true in infection age. If I consider what happens over the course of an infection, stuff that happens late in an infection is under weaker selection than stuff that happens early in an infection. And so in the context of COVID-19, for example, where we have most of the mortality happening late in the infection, even though it's selected against, selection against that should be very weak compared to selection for enhanced transmission early in an infection. And so last April, Sally Otto and Sylvain Gandon and Seb Leon and I published a paper in Current Biology where we started to explore the potential evolutionary consequences of what we might expect to happen during this pandemic. So remember, this was back in April before there was really any sense of any significant genetic variation in terms of in a functional sense, I should say. And again, I'm just going to very briefly mention a couple of these results. So one thing we looked at was basically the evolution of the timing of transmission. So there isn't enough data to be able to build that kind of a sophisticated model like I mentioned previously that Nicole was using. So we have this slightly simplified version of a compartment model where we have susceptible hosts, those that are infected but not yet transmitting and not yet symptomatic, those that are transmitting but still not symptomatic, and then those that are transmitting and are now displaying the mortality. So the mortality happening later. So you can kind of think of as infection age as progressing this way, basically. And then we asked questions about the evolution of different life history traits of the disease life history traits. So when, the pre-symptomatic phase, how would we expect that to evolve? The previous sort of results I showed suggested that in an epidemic scenario, there's a premium placed on getting lots of transmission out early in the infection, even if that comes at the expense of transmission later in the infection. I'm not going to speak too much to that. What I want to talk about more, given that's what's happening recently with COVID-19, is one of the scenarios we looked at where we imagined a variant appearing that was both more transmissible and more virulent. And so in this plot, the black line here is the frequency of this variant over time. The blue curves and the red curves are epidemiological dynamics. Don't worry so much about those for the present purposes. In this particular model, you can see this variant spreads through the population relatively quickly. And that's because transmission is under strong selection at the beginning of the outbreak, since there are lots of susceptible hosts around. And the cost of killing a host is relatively small, given that the mortality happens quite late in the infection, and using these sort of ideas from the evolution of senescence. The other thing to notice in this plot is that, so at the time we published this paper, it wasn't clear actually whether we were going to even be able to control the spread of COVID-19 in some areas. Now, of course, we know we can, but the sort of naive assumption we made at the time was maybe what we'll do is put strict lockdown into place and then turn it off and turn it on and turn it off. So these gray regions are periods of time where strict lockdown was on, and the white regions are periods of time where there was no lockdown. And what you'll notice is the evolutionary dynamics slow down during the periods of lockdown. And again, that's for the reasons that I described previously. During these lockdown periods, there's a reduction in the effective density of susceptible hosts. That reduces the selection coefficient on this variant that's more transmissible. And so its spread slows down during those gray periods. And I'm going to come back to that a little bit later on. Okay, so what about SARS-CoV-2? As everybody knows, there are now important variants called variants of concern that do have functional significance. And I'm going to focus only on this one on the left here, B117, sometimes called the UK variant. That's a variant that we now know is both more transmissible and imposes a greater degree of virulence. And I'll just show you a bit of data from this nice paper by Davies et al. That was published recently in Science that analyzes the spread of this variant in the UK. There's a whole bunch of analyses that they've done. I'll just show you a couple bits of them. So these are plots in 9 different regions in the UK, showing the relative abundance of all of these different variants shown on the bottom here. And this VOC one, that's what's known as B117 in other terminology. And the basic thing to see from this is there's lots of variability in these variants for a long period of time, but basically towards the end of last year and into the new year, this B117 variant essentially displaced everything else, suggesting it was really strongly favored at that point. And there are lots of different ways to estimate that selective advantage, and Davies have gone through a number of different analyses of that. This is just one of them, showing the sort of relative growth rate of the different variants and where they appeared in different times. And you can see that the variant of concern B117 has a considerably elevated growth rate or relative growth rate. And it's pretty unambiguous now that it's selectively favored because we've seen the spread, the selective sweep of this variant repeatedly and independently in many different geographic locations. Of course, the selective advantage of that variant could come from any number of different mechanisms. And so Davies et al. Also looked at different possibilities along those lines. So maybe it's increased transmission, maybe it's differences in duration of infection, difference in the kind of people that are infected and so on. Just to cut a long story short, the basic take-home message was that it appeared that an enhanced transmissibility was really the best explaining factor in terms of where this thing is gaining its selective advantage. In a separate paper, Davies et al., and then there's another paper by Challen et al. Here in the British Medical Journal, looked at the virulence consequences of this variant. And it's just a couple of snapshots from those two papers. These are survival curves. The top curve is for the wild type, if you like, and the bottom curve is for this B117 variant. Same ordering in both of these plots. And you can see that there is a significant change in the virulence, the mortality that's experienced by individuals that are infected with these different variants. So if we return to this plot that I showed you of this model that we were analyzing last April for a variant that has enhanced transmission and enhanced virulence, like we think B117 does, I want to now return to this idea that the selection coefficient is reduced during these periods of lockdown. Of course, what really happened is not an on-off lockdown, but instead a more graded kind of, rheostat kind of turning up and down of the lockdown. And this gray along the horizontal axis here is a reflection of that. It's a measure of strength of lockdown. It's the Oxford Stringency Index, where darker gray corresponds to a stronger lockdown. And this is for a particular region in England in this case. This is the frequency of B117 over time. And so if the predictions of this model are true, we should be able to go into this data set and look at the selection coefficient at any point in time, at any one of these weeks, for example, and see if it's related to the strength of lockdown at that time. The expectation being that the selection coefficient should go down as the strength of lockdown goes up because the availability of susceptible hosts goes down. And so we looked at that in 9 different regions from data in the UK, and you can see a pattern that's pretty suggestive of that. So there's no statistical analyses here, but this is a plot of the selection coefficient of B117 plotted against stringency index over the period of time when the selective sweep was happening in these different regions. And you can see a pretty consistent negative relationship between those two variables. Another thing you can do is you can look at ideas from evolutionary biology involving mean fitness and Fisher's fundamental theorem to make some predictions and see if those predictions are borne out in the data. So in particular, one thing you can do is you can suppose that if we look at regions where very little change in the lockdown was happening during the period of the selective sweep. So this data from London, for example, during this selective sweep of B117, the change in gray here is relatively minimal. If that's true, if we look in regions where that's true, then we should be able to make predictions about the relationship between the mean fitness of the population and the frequency of B117. Mean fitness being the slope of the epidemic curve here, the per capita rate of change of infections. And in particular, the mean fitness should be the wild type fitness plus the selection coefficient times the frequency of B117. And we have estimates of both S and P here, so we should be able to ask whether or not mean fitness as measured by this slope is linearly related to the frequency at each point in time with a slope equal to S. So that's the first prediction we can make in terms of mean fitness. Another prediction we can make is basically Fisher's fundamental theorem. So if we ignore any sort of fitness determining interactions among individuals, so we're ignoring things like herd immunity, for example. And again, if we're focusing only on cases where the stringency hasn't changed much over the course of the selective sweep, then Fisher tells us that we should expect the rate of change evolutionarily in mean fitness should equal the genetic variance in mean fitness. And so we can compute the rate of change of mean fitness as the curvature of this epidemiological plot. It's the rate of change of the slope. And we can compute an estimate for the genetic variance because we know the allele frequency or the variant frequency and the selection coefficient. So I'll just quickly show you those predictions and how they are borne out. So I've plotted here against B11 frequency, mean fitness, and these dashed lines are the predicted relationship of mean fitness as a function of B11 frequency. The curve is the estimate of mean fitness from the data. And you can see, if you squint at least in these two regions, it sort of roughly follows more or less the kind of increase you'd expect, but eventually it turns around. And even though the population is still adapting here, the mean fitness is declining, probably because of this enhanced stringency that's starting to occur late in the infection or late in the sweep, and possibly also from some enhanced level of herd immunity at that point. Fisher's fundamental theorem prediction, we can look at that as well. This dashed line is the estimated genetic variance during the selective sweep. It'll be highest when the frequency of the variant is at 50%. And the blue line here is a plot of the rate of change in mean fitness evolutionarily during this selective sweep. And again, if you squint a little bit, maybe you can kind of see a hint of what you might expect to happen, increasing initially, but then it decreases sooner than is expected theoretically. Again, presumably because of this enhanced stringency that's happening towards the end of the sweep, as well as maybe some level of herd immunity. Okay, so I'm just going to finish up quickly now with a couple of slides to ask the question about how important has evolution been in COVID-19 and what sorts of ideas might it offer us to help us control the spread. And when I mean how important has it been, of course, it's been important, and we know that's true because we've measured its selection coefficient. We know how it's changed R naught, and we know how that has affected our ability to control its spread. Another way you can ask this question is to go back into the UK data where the spread is already complete, basically, and ask, what would the epidemic look like if the spread of B117 had not occurred? So I want to go back into the data set, keep everything exactly as it was in terms of how stringency changed and all of that, but just essentially change all of the B117 infections to wild type infections and project forward over time to see what the outbreak would have looked like. And one way we can do this theoretically is, again, because we know mean fitness is the wild type fitness plus the selection coefficient times the frequency of B117, we can go in at any point in time in the epidemic and we can subtract off the selection coefficient times the frequency of B11 at that point in time to get a measure of what mean fitness would have been if all the infections were not B117, and integrate that forward over time to see what the outcome would be for the epidemic curve. And we've done that for a high and a low estimate of the selection coefficient in 9 different regions in the UK. So the top curve here and the dots are data for these different regions. The top curve is a fit to that data. And the gray region is an envelope of the projected expected outcome that would have happened in the absence of the evolution or spread of B117. So with a high estimate for the selection coefficient and a low estimate for the selection coefficient. And you can see that, with some areas, there's been an enormous effect in the sense of, if you look at the difference in the peak number of infections that happened with and without evolution, it's huge. Whereas in other areas, the effect is relatively minor. The last thing I want to just briefly mention is what we might use from evolutionary biology to help us understand and control the infectious disease. And again, this sort of harkens back to this paper we published in April, where one of the criticisms we got was, first, of course, there was no evidence for adaptive evolution at that time. But more importantly, even if there was, the criticism was that, would that really change what we do anyhow in terms of how we control the infectious disease if we knew about evolution? And so we addressed that by coming up with what I think we thought were a number of interesting ways you might think about how evolution could alter our control strategy. It turns out there's a really simple, almost trivial way that being evolutionarily formed is important that we didn't think of, and I don't know if we could have thought of, but it's turned out to be far more important than any of the things that we talked about, at least here in Ontario, Canada. And I'll just quickly explain that. So here's a plot of our weekly case count in Ontario from early December through to last week. And like many places, probably what we've done here to control it is, as we watch our curve go up like this, we start to get more and more freaked out. We impose stronger and stronger measures until the thing turns around and it starts to go down. And then as it goes down, we feel better about ourselves and we start to relax all of these restrictions, and then it turns around and goes back up. So we're always a little bit behind. We're always reacting to what's happening rather than somehow being proactive. Now, we knew at this point, given all of the data that came out of the UK, this decline that was happening throughout January in Ontario, we knew based on estimates of what the frequency of B117 was in Ontario, that there was something bad underlying this downward curve. And if you look at the frequency of B117 as it changed over that course of time, being evolutionarily informed, we knew that despite the fact that this was going down, there was an underlying outbreak of B117 that we were not controlling. And it was eventually going to come back to haunt us unless we were somehow proactive in not releasing all of our control measures on this downward turn here. And so taking that evolutionary perspective allowed us to strongly advocate that even though things look good, they're not good if you look at what's happening in terms of the evolutionary change in the composition of the pathogen population. Unfortunately, that didn't actually sway many people who were making decisions at the time. So we did release, and eventually things kind of went back up again. And this upswing here is due almost entirely to the spread of B117 now, because it's become the dominant variant in Ontario. Okay, so I'm going to stop there. I'll just put up this last slide of acknowledgements. There have been a large number of people over the years that I've worked with doing all kinds of things along these lines, and I want to hopefully make sure I remember to put everybody up on this slide, and I'd be happy to take any questions anybody has.
Thanks so much, Troy. Very interesting. We're already getting some questions in. I can start with one of my own.
Sure.
So, obviously, I guess the genetic covariance between transmission rate and virulence is very important, right?
Yep.
And so, what is the the evidence of what this correlation can be? Do you know, like in COVID or maybe in influenza?
Yeah, so that's been a subject of quite a lot of work over the years because it sort of forms the basis of what's called the trade-off hypothesis for the evolution of virulence. That hypothesis being based on an assumption that there's a positive covariance between transmission and virulence. And so there's been a lot of work devoted towards trying to see whether or not that's true in different systems. There is in some systems and there isn't in other systems. I mean, there are some mechanistic reasons to expect that relationship to be there. Maybe, high replication rate results in higher transmission, but also greater mortality, or maybe a higher replication rate stimulates the immune system more, and it's the immune system that imposes a lot of the mortality in some infections. But it is a bit variable from one infectious agent to the other. In the case of COVID, we don't really know at this point. So it is true, it seems from the data, that the new variant B.1.1.7 has higher transmission and higher virulence. That could just be some shitty random luck that we've had, and it's not a general negative or positive correlation between those things genetically. It might just be a chance event for that particular variant. At this stage, it's not known.
I guess any decoupling would be heavily favored, right?
Yeah, exactly. So if we have another variant that's equally transmissible but a lower virulence, then it would certainly spread. Although the problem with COVID-19, as I kind of alluded to, is that the cost of being virulent is pretty tiny, since most of the mortality that happens late enough in the infection that it doesn't have a lot of consequences in terms of the pathogen's fitness.
Right, great, thanks. Someone asked whether you could talk a little about the age structure of the host population and how it can influence disease emergence and interact with pathogen transmission.
Yeah.
For example, where host susceptibility is positively correlated with increasing host age, that is, older hosts are more susceptible than younger hosts, how could this be included in your models?
Yeah, that's a great question. So it's, I mean, as you could imagine, there are a number of different ways that could pan out depending on the sorts of assumptions or what's known about that relationship. One way to think about what it does is it sort of, you can think of the host population as a resource. And in the simple models that I've looked at, or I've presented here at least, there's essentially just one resource that the pathogen is using. And if we now structure the host population, maybe by age or any number of other things, immunological history, then there's a suite of different kinds of resources that are present in the host population. And so that allows for, to some extent, maybe not surprisingly, greater diversity of pathogen strategies possibly as well. But it can also affect emergence in the way I guess you're thinking, because it will, you could have greater or lesser degrees of susceptibility to infection, greater or lesser degrees to which you transmit the pathogen. Maybe different age groups have higher connectivity than other age groups. There has been some work looking at contact matrices, basically. So how does the contact pattern between people of different ages play out? Not surprisingly, there's lots of contact between working aged people. There's contact between young people and working age people who are, the kids of those working age people. And then there's much less contact often among other age groups. So, I mean, it really does depend on how all those things intermingle, but it's certainly possible. And there have been some analyses that look at that.
Yeah, so social network analysis
Yeah,
is like
exactly.
a hot thing, right?
Exactly, yeah.
If transmission at early stages trades off with transmission at later stages of infection, I guess this suggests that there are different traits, that these are different traits. Otherwise, the same genotype would transmit more or less at these two time points, right? Is there evidence that these are different things?
So I should stress that that pattern was specific to those genotypes, first of all, and that specific pathogen. So whether or not that's a general general relationship in other pathogen species is not really known, but you're right that they are different traits in a sense. But the one way to think again about mechanistically what's happening, and I think that's probably what's happening in this particular case, is if you replicate and sort of expose yourself to the immune system very quickly early on in an infection, you get transmission early on, but then the immune system comes online and you get killed off quickly. If you wait, you can have that transmission, but you have it later. So there's a sort of an interaction between when you expose yourself to the immune system and where the transmission is happening. I mean, we've built mechanistic models that try and capture that thing, and you do see that very same pattern of negative genetic covariance coming out of that for certain assumptions about how the immune system is triggered as a function of when the pathogen starts to replicate or the rate at which the pathogen replicates.
Great, thanks. So one thing we haven't talked about too much is vaccines. So in the context of age-based vaccination rollouts, how will selection for transmission in kids on one hand and immune escape on the other hand compare, do you think?
That's a tough question. So, I mean, it's an important question, obviously, and I think we're bound to see evolutionary change in COVID as a result of vaccination. So, there are a couple of, I mean, I'm not going to have a satisfying answer for you, unfortunately. There are a couple of things uh, to think about, though. One of them is the immune escape idea, for sure. You know, variants that are able to escape the immune system somehow will certainly have a selective advantage. Uh, the other thing, though, and I meant to mention it and didn't, is in that general sort of analysis I talked about where the density of susceptible hosts, when it's high, it selects for higher transmissibility. That same process plays out in the context of a vaccine rollout as well, because again, if you think of it as different resources, before the rollout, there's essentially, in the simplest world, a single host population, which is a single resource, and then that resource is kind of depleting as herd immunity is coming in. But when we start to vaccinate, that then generates a diversity of resources in the population from the pathogen standpoint. It generates some hosts that are vaccinated and are a different resource in some sense, and those that aren't. And so you can imagine the very same kinds of processes happening, because now you're generating a very large density of susceptible hosts. When I say susceptible, I mean those are susceptible to a vaccine escape mutant, right? So you have access to this huge pool of susceptible hosts all of a sudden. And that very same dynamic that we're sort of thinking is playing out right now in terms of the evolution of enhanced transmission could play out exactly in the same way in the context of vaccinated populations and for the very same kinds of reasons. So, I mean, that's not an answer to your question, but I think there are many things that we need to think about in conjunction with one another like that. I know there are a number of models people are building right now to try and explore these different possibilities, but I think the other thing that is worth stressing, though, is probably the most dangerous thing right now that we're doing is vaccinating some people and then having them intermingle with people who are infected and not vaccinated, because that basically increases the pool of new potential variants that can come in and potentially infect vaccinated individuals. It sort of increases the mutational supply, if you like. And so, I mean, there's very little I think we're going to be able to do to stop that. But in an ideal world, we would do things where we'd sort of try and separate in some way those individuals that were vaccinated from those that weren't, so that we didn't get any sort of cross-group transmission that might enhance the evolution of vaccine escape. That's a pretty tall order, though, for any sort of realistic intervention strategy, I think.
Yeah, so related to that last bit, there's also the question of whether to, in the two-dose vaccine, right, whether you want to give everyone their first dose as quickly as possible or try to get 2 doses in everyone as quickly as possible. Does that affect the mutation chances?
Yeah, that's important as well, and it's important at least in a few ways. One way is it depends on whether or not, so when you need 2 doses, if you need 2 doses because the first dose sometimes doesn't work, that results in a different sort of selective environment than if it was a situation where the first dose always works, but only halfway, and then the second dose takes everybody to the full immunity, right? So those are sort of 2 extreme possibilities. One is that the vaccine is working more or less in everybody, but you need 2 doses to get everybody up to full immunity. The other is that you only need 1 dose. You sometimes get full immunity with one, and sometimes you don't. And the second dose is trying to get all those that it didn't work in the first time. those are very different selective environments in terms of what we might expect to happen, because in the case where you have a half level of immunity, there's a sort of environment there where you might imagine it favoring the emergence of vaccine escape mutants more readily than in a case where you had either 100% immunity or none at all.
Right, thanks. Someone asked, what are your thoughts on South African and Brazilian VOCs coming to Ontario?
Yeah, I mean, they're here. So they have certainly, I mean, there's good evidence of their spread, obviously, in some places. The data for the selective advantage of them is, I almost say, less compelling. It's just, I don't think we have enough of it yet. And they may be just as bad as the UK variant, and we just don't have quite the quality and amount of data that we have for the UK variant. It's also a little bit unclear how, if they are selectively advantageous in similar ways, and they do have suites of mutations that would suggest they are, how the competition between those and the UK variant play out. I think that's a big unknown, and it's something that we're going to probably see unfold in the next very short while.
Right, the more general question to finish off then. If the cost of virulence can be ameliorated by killing at late infection age, might we expect zoonotic pathogens that successfully jump from distantly related hosts, where maladaptive virulence is a bigger issue, to cause mortality at a later stage than those that jump from more closely related hosts?
So the reasoning being that if it's from a distantly related host, it is not as adapted to the new host, and so it has a higher than equilibrium level of virulence, I guess, is the idea. And somehow, if it's late in an infection, that can be sustained. I suppose that's a possibility. I mean, it's certainly true that if you consider all possible cross-species transmission events, those that have lower virulence or those that have high virulence, but it's expressed late in an infection, are going to have a higher likelihood of actually taking hold and being established in the new population. For the same sorts of reasons that they're just, it's a relatively unimportant component of the pathogen's fitness if it happens late enough in the infection.
All right, I think that's where we'll leave it today.
Okay. Thank you very much.
Yeah, thanks again, Troy. Next week, as I said before, we'll have Anil Agrawal, and I hope to see you all then. And until then, stay safe. Goodbye.